Think of your immune system as a ruthless, microscopic apex predator. Every second, millions of these cells patrol your bloodstream, actively hunting down viruses, bacteria, and anything else that doesn't belong. But this massive destructive power comes with a severe vulnerability. If these biological weapons make even a tiny calculation error, they can misidentify your own healthy tissue as an invading enemy. This friendly fire is the mechanism behind autoimmune diseases. When the body launches a war against itself, it begins destroying the skin, the organs, and the joints, leaving lifelong damage. For decades, medical science thought it had an answer for this. Researchers believed the body had a quality control system in an organ called the thymus, where any defective, self-attacking immune cell was deleted before it could ever enter circulation. Except, clinical evidence eventually proved that dangerous, defective cells were routinely surviving that quality control process and slipping out into the bloodstream anyway. If lethal, miscalibrated immune cells are constantly escaping into the body, every human being should, in theory, be continuously self-destructing. In 1995, a Japanese researcher named Shimon Sakaguchi stepped forward to address this biological contradiction. He proposed a hypothesis that went against the established textbook science. He argued that the body deliberately manufactures a specialized class of immune cells whose only purpose is to hunt down and stop other immune cells. Instead of attacking foreign invaders, these cells act as emergency brakes. If a rogue immune cell attacks healthy tissue, these microscopic peacekeepers mechanically intercept and shut it down. Sakaguchi named them regulatory T cells, or simply T-regs. But despite his experimental evidence, the broader scientific community remained skeptical. Proving they existed wasn't enough. Nobody knew what biological mechanism actually created or controlled these peacekeepers. To turn this observation into actual medicine, researchers needed to find the exact genetic source code that built a T-reg cell from scratch. Around that same time, American researchers Mary Brunkow and Fred Ramsdell were trying to understand a horrific anomaly in a specific lineage of mutant mice. These mice suffered from catastrophic system-wide inflammation. Their organs failed almost immediately as their entire immune system spiraled into total self-destruction. Mapping the genome to find the root cause, researchers isolated a single mutated gene sequence, the FOXP3 gene. It's the master biological switch for generating T-reg cells. Without it, the body cannot build any biological brakes, allowing immune cells to go rogue. The same mechanism operates in humans. Children born missing this specific gene develop a rare and devastating condition called IPEX syndrome, where their immune systems attack nearly every organ in their bodies. This single gene, and the army of T-reg cells it produces, is an absolute, non-negotiable requirement for human survival. By mapping this entire pathway, these scientists earned the 2025 Nobel Prize in Medicine. They took a biological observation and turned it into an active medical toolkit. By artificially boosting the production of T-regs, doctors may soon treat autoimmune diseases at their root cause, or teach the body to peacefully accept transplanted organs without relying on toxic immune suppressants. Conversely, cancer tumors often exploit these T-reg cells to hide from the immune system. Selectively disabling those specific biological brakes would allow the body to attack tumors with far more aggression. Modern medicine is moving toward mastering the microscopic peacekeepers we already possess, finally learning how to stop the body